Hello everyone, myself Dr. Tanushri Chatterjee from Raipur Institute of Technology. Welcome in the SWAYAM course of Fundamental of Computational Biology. Today I will discuss about molecular switch and its application in the field of biotechnology. So, what is molecular switch? Molecular switches are essential components in the field of nanotechnology and molecular biology, playing a pivotal role in the regulation and control of various biological and chemical processes. These switches operate at the molecular level, allowing for precise control and manipulation of molecular functions, making them a crucial tool in modern science and technology. If we see the historical perspective of molecular switches, the history of molecular switches can be traced back to the early discoveries and pioneering work of scientists who laid the foundations for our understanding of molecular behavior and reactivity. Over the year, researchers have made significant contribution to elucidate the principles governing molecular switches, shaping the field of molecular science and setting the stage for modern advancement. One of the earliest observations related to molecular switches dates back to the 19th century, when scientists began exploring isomerism. Isomerism refers to the phenomena where molecules have the same molecular formula, but differ in structural arrangement or spatial orientation. Archibald Scott Cooper and Alexander Berlow independently proposed the concept of chemical structure and isomerism in the early 1860s. This breakthrough was fundamental in understanding how subtle structure changes in a molecule can result in different chemical and physical properties. In the late 90s and early 20th centuries, chemists like Jacobs von Hoff and Alexander Bernoulli among others made crucial contribution to stereochemistry and conformational isomerism. Von Hoff research on asymmetric carbon atoms and spatial arrangement of atoms laid the foundation for our understanding of molecular asymmetry, which is key to many molecular switches. Later, in the mid 20th century, advances in the spectroscopy and quantum chemistry provided deeper insight into molecular behavior. The advent of X-ray crystallography by Sir William Henry Bragg and Sir William Lawrence Bragg in the early 20th century allowed scientists to determine the three-dimensional structures of molecules with high precision. In the 1950s and 1960s, the development of quantum chemistry and computational methods by pioneers like Robert Parr, John Pople and Walter Kohn provided theoretical framework for understanding molecular properties and electronic transitions. These developments paved the way for a more systemic and theoretical exploration of molecular switches. the field of molecular switches gained significant momentum with the discovery of photochromic molecules, particularly azobenzene by Rolf Husgen in the 1950s. Azobenzene demonstrated reversible photoisomerization, switching between trans and cis isomers upon exposure to light. This discovery laid the groundwork for photochromic molecular switches, which are now extensively utilized in various applications. In the 1970s and 1980s, researchers like Fraser, Stoddart, Jean Pierre Sauvage and Bernard Frejnga made groundbreaking contribution to the field of molecular switches through the development of mechanically interlocked molecules, including rotazenes and catenanes. These molecules exhibit control movement and conformational changes resembling molecular level machine or switches. Now, why we study molecular switches in biotechnology? So, studying molecular switches in biotechnology is essential for several reasons, primarily due to their pivotal role in manipulating and controlling biological processes at the molecular level. Molecular switches serve as valuable tools for understanding, regulating and designing novel application in the field of biotechnology. First one is precise control of biological process. Molecular switches enable precise control over biological events such as gene expression, protein activity and cellular signaling. This control is crucial for studying the intricate mechanism within cells and organisms, providing insight into disease pathways and potential therapeutic targets. You can see in this pictorial representation various field. Then second one is the drug development and targeted therapies. Understanding and utilizing molecular switches is fundamental in drug development by designing drugs that act as molecular switches. It is possible to target specific biological molecules or pathways, improving drug efficacy and reducing side effects. This precision allows for the development of targeted therapies tailored to individual patient. Third one is the diagnostic tools and biosensors. Molecular switches can be incorporated into biosensor and diagnostic tools, facilitating the rapid and sensitive detection of biomolecules. This is pivotal in medical diagnostic, enabling early detection of diseases, monitoring treatment response and assessing overall health. Fourth one is the smart drug delivery system. Utilizing molecular switches in drug delivery system allows for control and targeted release of therapeutic agents within the body. These systems respond to specific triggers such as changes in pH or temperature, ensuring drugs are released at the right time and in the right location, enhancing therapeutic outcomes. Fifth one is the biological imaging and visualization. Molecular switches play a significant role in advanced imaging techniques, allowing scientists to visualize and track specific biomolecules in real time within living cells. This is vital for understanding cellular dynamics, localization of proteins and studying disease progression. Sixth one is the synthetic biological and bioengineering. Molecular switches are key components in synthetic biology, enabling the construction of synthetic genetic circuits and systems. This capability is essential for developing novel biotechnological applications, including biosynthesis of valuable compounds, metabolic engineering and biofuel production. Another type is the environmental monitoring and remediation. Molecular switches can be employed to develop biosensor for environmental monitoring, detecting pollutants, toxins or pathogens. This contributes to environmental sustainability by enabling early detection and efficient remediation strategies. This remedy you can see here. Next one is biological computing and information processing. Molecular switches can be harnessed to create biological computing system and information processing devices. This open up exciting avenues for bioinformatics, data storage and computing paradigms that use biological molecules to process and store information. Now, landmark discoveries in switches. Landmark discoveries in molecular switches are azobenzene and photochromic switching. A seminal discovery in the field of molecular switches was the photoisomerization of azobenzene reported by Rolf Husgen in the 1950s. Azobenzene, a simple molecule with two phenyl rings linked by a central nitrogen bond, demonstrated reversible isomerization between trans and cis configuration upon exposure to light. This discovery opened the door to photochromic molecular switches, enabling precise control over molecular structure and functioning, uses light as a stimulus. Rotagens and catenanes. The development of mechanically interlocked molecules such as rotagens and catenanes marked a major milestone in molecular switch research. Fraser Stoddart, Jean Perry, Sauvage and Bernard Feringia played key role in this breakthrough. Rotagens consist of a molecular ring trapped onto a axle along controlled movement of the ring along the axle. This molecular architecture paved the way for the creation of molecular machines, providing insight into molecular motion and allowing for the design for molecular switches with controllable functionalities. Spiropyrons and di-area lithians for molecular photo switches. In the 1970s, researchers discovered the light induced isomerization property of spiropyrenes and di-area lithians. These molecules undergo reversible transformation between closed and open forms upon exposure to light, making them valuable in designing molecular photo switches. The ability to toggle between states by light activation has been pivotal for application in molecular electronics, data storage and biological imaging. Next one is the fluorescent protein and optogenetics. The discovery and the development of fluorescent protein such as green fluorescent protein GFP and their application in optogenetics revolutionized the field of molecular switches in biological research. Optogenetics utilizes light sensitivity proteins to control the manipulate cellular processes with exponential precision. Nobel laureate Osamu Shimomura, Martin Kalfe, Roger Whitesane made significant contribution to the discovery and utilization of fluorescent protein, enabling non-invasive monitoring and control of biological activities at the molecular level. Next one is dynamic covalent chemistry and constitutional dynamic chemistry. The introduction of dynamic covalent chemistry by Jean Marie Lehn in the 1980s was a paradigm shift in molecular switches. This approach involves the reversible formation and the breakage of covalent bond, allowing for dynamic structural changes and switchable functionalities. It laid the foundation for constitutional dynamic chemistry, providing a new perspective on molecular rearrangements and stimuli responsive behavior. You can see in this picture. Now, DNA nanotechnology and DNA based switches. This advent of DNA nanotechnology and the development of DNA based switches expanded the scope of molecular switches. DNA with its predictable base pairing serve as an excellent scaffold for constructing complex nanostructures with switchable properties. This innovation, pioneered by Ned Seaman and others, has potential application in drug delivery, diagnostic and nanoelectronics. Mechanochromic and piezochromic material. The discovery of mechanochromic and piezochromic materials which change color or fluorescence in response to mechanical stress or pressure has opened new avenue for molecular switches. These materials exemplified by spiropyranes and diarylethanes have applications in sensor, smart material and stress responsive devices. Now, what are the types of the molecular switches? The versatility of molecular switches makes them invaluable for numerous applications including drug delivery, sensor, information storage, molecular machines and many more. First one is photochromic molecular switches. Photochromic switches undergo reversible structural and conformational changes in response to light, typically in the UV or visible range. The two main stages are closed and open forms, which can interconvert open exposure to light. Azobenzene, spiroranes, diarylethanes and fulgides are well known photochromic molecules. Now, azobenzene. Azobenzene switches between trans and cis isomer upon exposure to UV or visible light. This property has application in nanotechnology, drug delivery and optical devices. Spirorens and diarylethenes, spirorens and diethylenes can transition between non-polar, that is closed, and polar, that is open forms under light, showcasing potential in molecular electronics and optical switches. Next type is the pH sensitive molecular switches. pH sensitive switches respond to changes in acidity or alkalinity by undergoing structural or conformational alterations. They find extensive application in drug delivery, biosensing and controlled release systems. Hydrogen based switches. Hydrogen bonds can be cleaved or formed based on pH changes, making them suitable for pH responsive drug delivery system. Next type is azo based switches. Azo dye can undergo pH induced isomerization, allowing their use as pH sensor and drug carriers. Next type is the temperature sensitive molecular switches. These switches respond to changes in temperature, triggering alterations in their structure, solubility or properties. This property is exploited in smart materials and controlled drug delivery. Polyanisopyrylacrylamide or PNI-PAM. This type exhibit a lower critical solution temperature around body temperature, making it a suitable candidate for thermally triggered drug release system. Fourth type is electrochemical molecular switches. Electrochemical molecular switches are responsive to changes in electric potential or current. Redox active molecules are commonly used in this type of molecular switch. Redox active switches, in these molecules with reversible redox states can switch between different oxidation states, providing potential applications in molecular electronics and sensors. Fifth type is mechanically responsive molecular switches. This type of switches respond to mechanical force such as stretching or compression, causing structural changes in the molecule. This category includes mechanochromic and piezochromic materials. Mechanochromic materials. So, what are the mechanochromic materials? These molecules change color or fluorescence in response to mechanical stress, and they find application in sensors and stress responsive devices. In piezochromic materials they exhibit changes in optical properties under pressure, useful for applications in pressure sensors and material science. Sixth type of molecular switches is chemically responsive. Chemically responsive molecular switches. Chemical stimuli such as the presence of specific ions or molecules can trigger structural or conformational changes in these switches. They are used in chemical sensors and diagnostics. Ion sensitive switches are switches change their properties in the presence of specific ions, making them useful for ion sensor and biological applications. Seventh type is DNA based molecular switches. DNA can serve as a versatile platform for designing molecular switches due to its predictable base pairing and structure. DNA switches have applications in drug delivery, biosensing and nanotechnology. DNA nanotechnology. So, what is DNA nanotechnology? DNA based switches can be engineered for precise control over molecular functions and have implications in targeted drug delivery and gene regulation. Eighth type is optically controlled molecular switches. These switches are activated or controlled using light, often through optically active molecules or photosensitive groups. Photoisomerizable switches. So, molecules capable of reversible isomerization under specific light wavelengths, offering application in molecular electronics and light control systems. Ninth type is molecular logic gates. Molecular logic gates are constructed using combination of molecular switches to perform logical operations akin to electronic logic gates and are crucial in molecular computing and information processing. And or NOT gates, combination of molecular switches can perform logic operations contributing to the development of molecular computing and information storage. Next type is multi-state and multifunctional molecular switches. These switches can exist in more than 2 states and possess diverse functionalities, providing enhanced versatility and complexity. Multi-state switches are molecules capable of multiple reversible transformations, allowing for a wide range of controllable states and functionalities. Next type is metal organic frameworks as molecular switches, that is MOFs, metal organic frameworks. With their tunable structures and properties can act as a molecular switches or host molecular switches within their pores. So, MOF based switches can be designed to encapsulate or incorporate molecules with switchable properties, broadening their potential application in various fields. Next type is biochemical and biomolecular switches. Biological molecules and biomolecular complexes can also function as molecular switches responding to biological signals or interactions. So, protein based switches are, these protein can act as a molecular switches, changing conformation or activity in response to ligand binding, pH or temperature. This is fundamental in cellular signaling and regulation. Now, design and engineering. So, rational drug designing strategies. Rational designing strategies refer to a systemic and deliberate approach in designing molecules, materials or systems with specific desired properties or functionalities. Understanding structure, property, relationships. A fundamental aspect of rational design is understanding the relationship between the structure of a molecule, material or system and its resulting properties. Second one is computational approaches and modeling. Computational tools and techniques play a crucial role in rational design. Computational chemistry and physics simulations, molecular dynamics, quantum mechanism calculations and machine learning algorithms can provide valuable insight into molecular behavior and interactions. Third one is quantitative structure activity relationships. Quantity structure and activity relationship is a mathematical modeling approach that correlates the structural features of molecules with their biological or chemical activities. Bottom up and top down approaches. So, what are the bottom up and top down approaches? Rational design employs both bottom up and top down strategies. Bottom up involves building complex structure by assembling smaller, well defined building blocks. Next one is biomimicry and bio-inspiration. Nature often serves as a rich source of inspiration for rational design. Studying biological system, their structure and mechanism can guide the creation of synthetic material or devices with biometric properties. Sixth one is modular design and building blocks. Modular design involves creating a system or material using standardized building blocks or modules. These modules can be assembled in various combinations to achieve different functionalities, making the design process efficient, flexible and adaptable. Next one is feedback loop and iterative design. A critical aspect for rational design is the interactive process that involves continuous feedback and refinement. Initial designs are evaluated, under the insight gained are used to improve subsequent iteration, gradually converting towards the desired properties and functionalities. Next one is material genome initiative. The material genome initiative leverages high throughput, experimental and computational methods to accelerate materials discovery. Next one is directed evolution in biology. In biotechnology, directed evolution involves evolving biomolecules like enzyme or proteins to enhance or modify their functions for specific applications. 10th one is the additive manufacturing and 3D printing. Additive manufacturing technologies like 3D printing allows for the precise and customized fabrication of structure based on digital designs. So, what is directed evolution? Directed evolution and high throughput screening. Directed evolution is a powerful technique used to modify and optimize biomolecules, particularly proteins and enzymes, to enhance their property or introduce new functionalities. So, key steps in directed evolutions are mutation, diversity generation, selection or screening, amplification and alteration. So, application of directed evolutions are enzyme optimization, protein engineering and drug development. High throughput screening is an automated, rapid and efficient experimental process and used to evaluate the biological or biochemical activity of a large number of compounds or biomolecules simultaneously. So, key step in high throughput screening: library preparation, automated assay, data collection, data analysis and hit selection, and its applications are drug discovery, functional genomics and compound profiling. So, computational approaches in switch design is quantum mechanical and molecular dynamics simulation. So, this is quantum mechanical simulation and molecular dynamic simulations. Second one is ab initio calculation. ab initio calculations are performed without realigning or experimental data. Starting from fundamental principles, they are highly accurate and are used to predict molecular geometrics, electronic structure and energies. Third one is density functional theory. DFT is a widely used quantum mechanical method that calculates electronic properties of molecules. It provides valuable information on molecular structures, charge distributions and transition states, crucial for designing efficient molecular switches. Then Monte Carlo simulation. Monte Carlo simulation are probabilistic algorithm used to model the behavior of systems with random variables. Next one is core grained modeling. Core grained models simplify molecular structure representing group of atoms as single particles. This approach reduces computational complexity, allowing the study of larger molecular systems. Sixth one is machine learning and artificial intelligence, descriptive analysis and QSPR and QSAR models. Seventh one is free energy calculation, hybrid quantum. Eighth one is mechanical molecular mechanics. Now, application in drug discovery, I will give you some case study also. First case study is liposomal drug delivery system. In liposomal drug delivery system represent a significant advancement in the field of drug delivery, offering enhanced therapeutic efficacy and reduced side effects compared to conventional drug delivery methods. Second case study is, for example, Doxil. Doxil is a well known liposomal formulation of doxorubicin, an anti-cancer drug. Liposomal encapsulation of doxorubicin offers several advantages including prolonged circulation time, targeted drug delivery to tumor sites due to leaky vasculature and reduced cardiotoxicity associated with the free drug. Next case study is control release for cancer therapy. Control release for cancer therapy involves designing drug delivery system that allow for the precise, sustained and targeted release of anti-cancer agent. Another example is temozolomide. Temozolomide, it is a standard chemotherapy drug used for glioblastoma multiforme, a malignant brain tumor. So, you have seen some case study also, some examples also. I think it is clear to you. Thank you.